We are live, about to go live on Sparkle.
Hello, good morning, good afternoon, good evening. We're still missing Anna. So she should come as well. We were in a different room a minute ago. So can anyone hear me? Because I cannot hear anyone.
Yes, I can hear you, Andre.
Okay, okay, very good. Okay. And like I said, I was with Anna in a different room. Don't know how that happened because I thought I clicked the same link. So thanks for forwarding that, Ross. We are good to go. Oh yeah, okay. I see we're already there. So hello, good morning, good evening, good afternoon. My name is André Fischer. Welcome to the Siemens Healthineers Symposium. Give me a second to share my slides. So I'm the responsible marketing manager for EuroMR at Siemens, and we have, now I see we have everyone with us. I see that Anna Wang is now with us, and also Ross Mayer from Randy Buckner's lab. So these will be the two speakers we will have today in our symposium. So thanks for joining and welcome. And before we start, I will quickly say a couple of words about our what we have to offer in the context of advanced neuro research. I think I have to share my screen again. So I hope that looks better. So so the question is, what does Siemens has to offer in the context of advanced neuro research? We think we are well equipped there. So we have, on the one hand, the best tools in our opinion for advanced neuro research, the collaboration network, and with a 7T Magnetum Terra and the 3 Tesla Magnetum Prisma, two really outstanding systems. And then I want to quickly go in a little bit into how some of these research applications later on manifest itself as a product on our systems. But first start with a collaboration, because this is really at the core of what we do and what is very important to us to translate the power of MR research ideally into clinical care. And this of course is only possible if we do that together with you. And our global research and collaboration network is, with more than 400 sites, pretty strong, pretty big. We established a couple of years ago more than 1,100 new projects. And if you look at the whole company, Siemens Healthineers, then almost 1 out of 2 research projects that we started at Siemens Healthineers was focused on MR, which again outlines how incredibly useful and powerful MR is in the research. To make that happen, we have a lot of MR collaboration scientists out there in the Americas, Europe and Africa, and also in Asia and Pacific. So the message here should be that one of these scientists is always reasonably close to you and willing to help. And of course, you can always reach out to any one of us, be it in headquarters, be it someone you have already contact with in Siemens. So please reach out to us. We are there to help. We want to make it happen together with you. And we want to make it as easy as possible for you to collaborate with us. That means eliminating paperwork wherever possible, to make it also possible to exchange new research prototypes amongst each other. We call that C2P, Core Competence Partnership, which, like I said, in the end, just means that you can exchange your research prototypes with other sites in an easy way to facilitate broader research projects in that area. For that, you need, of course, the right hardware. We think with the Magnetum Terra and the Prisma, you're really, really well equipped. Especially the 7T, I mean, that goes into what is now called ultra-high field imaging. That goes back to 1999 at Siemens. A first commercial system was already installed in 2002 at MGH, and then things progressed and got more mature. We got also some installations with 9.4 Tesla and even 10.5 and 11.7 Tesla. So we're really dedicated in that area, and it all culminated in the Magnetum Terra that was unveiled 2015 and released for clinical use in 2017. But it does not mean that we stop there. We want to further drive it. So metabolic imaging, for example, with multi-nuclear option is of course very interesting at these high field strengths. And also building more dedicated hardware like the impulse gradient coil from the University of California in Berkeley. That's one of these projects. And like I said, we will continue to innovate in that area. So just because the Terra is out there, it does not mean that we stop anywhere. We talked a lot about the Terra. Here are some specs like the 8200 gradient system. As I already said, it's the first 7T system that was released for clinical use. It supports, for that reason, a dual mode, so a research mode and a clinical mode, supports up to 64 receive channels and is on the modern software. The Magneton Prisma also recently got a software upgrade, so this is why we call it now Accelerate Edition. You get a new software called XA30 that allows you to use things like MR fingerprinting, new compressed sensing applications. But the specs are still the same, of course, 8200 gradient system, parallel transmit architecture, and a large number of channels, either 64 or 128. So let's quickly talk about how to translate that, how that sometimes translates into clinical tools. And I mean, MR fingerprinting is a topic that we share for quite a while. And it is one scan, as most of you know. It delivers at the moment two parameter maps, the T1 and the T2 map. And the nice thing about it is that in principle you can add more parameters as long as you can model these parameters. And to make it easier also to tailor MR fingerprinting a bit more to your needs, we now have something called the MRF Development Kit. This allows you to have T1 and T2 ranges that you would determine. They come as an input. They create a new sequence. That sequence is then used to perform a Bloch equation to get a new dictionary for quantification. And both the dictionary and the new sequence are bundled into what we call the MRF container. That's just basically a container for these two things. And then you copy or transfer them to your 3 Tesla scanner, and then you can execute it there, given that you have a valid MRF license, of course. These systems have to run on XA30 or XA31, such as the Prisma Fit, the Skyra Fit, or the Vida at the moment. Then on the other hand, we have SMS. This is a clinical product for quite a while. This helps really tremendously with diffusion imaging and also advanced applications like diffusion tensor imaging or bold imaging to bring that more into clinical routine and also facilitate faster research, of course. And then there's also one thing that you can only get with us, with Siemens Healthineers, and this is the SMS TSE and also the SMS Resolve, so a readout segmented EPI sequence. This is just to highlight some examples. So for example, here for DTI, instead of the conventional 12 minutes, as you can see here on the left, you can cut down that scan time essentially by a factor of 3 by using SMS, which makes it clinically much more interesting. Then for research applications such as diffusion spectrum imaging, also you can cut down the scan time from here 45 minutes down to something in the 15-minute arena, also much more realistic and less prone to motion artifacts. And then of course for fMRI, this brings really substantial reductions in scan time. For example, here shown on a factor of 8, SMS factor of 8, that allows to use really low TR values and provides a method to sample the bold response with high spatial and temporal resolution. But now I don't want to eat up more time. We have two great speakers in that symposium, and I already talked about them. So on the one hand, we will have Randy Buckner, who will talk about precision estimates of structure and function within the individual. And on the other hand, we have also live with us here in the meeting, Dr. Anna Wang from Zhejiang University in China, and she will talk about INS fMRI, a method for mapping mesoscale connectomes in non-human primates. And this will be the talk that we start right away. As already said, Dr. Wang is with Zhejiang University in Hangzhou in China. She's the professor of biomedical engineering, neuroscience, and clinical neuroscience. She's also the director of the Zhejiang University Interdisciplinary Institute of Neuroscience and Technology, and also the director of the Neuroengineering Consortium. Her academic career started in Harvard. She did her PhD at MIT, then had various stations at Rockefeller University, Baylor College. She was with the University of Queensland in Australia. Then another position at Yale and Vanderbilt University, and since 2015 she's now with Zhejiang University. She's a fellow of AAAS and SPIE. She holds several editorial roles, for example, for Neuroimage. She's a member of the OHBM Abstract Review Committee and has a large number of peer-reviewed publications on PubMed, more than 100. And so we are really thrilled to have Anna Wang here with us. And now we will see or learn about her presentation about INS fMRI. Please, Dr. Wang.
Hello, my name is Anna Wang. I'm at the Interdisciplinary Institute of Neuroscience and Technology at Zhejiang University in Hangzhou, China. I'm going to talk to you today about INS fMRI. It's a new way for mapping connections in the brain in vivo at high spatial resolution or mesoscale. This technology was chosen as one of the top 10 medical technologies in China in 2019. So I'm going to talk to you about architecture of the brain. So one of the things we know that architecture is important. The IBM Blue Gene is a supercomputer which occupies 300 square meters, performs 10 to the 14th operations per second, and uses 1 million watts of energy. The human brain, in comparison, is 1.5 kilos in size, and it performs 10 to the 15th operations per second using only 20 watts of energy. So what makes this difference? Well, it may be the importance of its architecture that offers quantitative and qualitative advantages. So because of this, there are many connectome projects in the world that are studying the way networks are organized in the brain. And in China, there are projects at macro scale, at micro scale, and in our university at mesoscale. So what is new, or what is the architecture of the human brain? What is special? Well, the human brain and monkey brains share very similar behaviors. They share visual and hand behaviors that are similar, as well as social behaviors. In addition, their brain organization is quite similar. These columns, are so-called functional units, are systematically organized in the brain. These units are also developmentally determined, as shown by Pashko-Rikish, where there are protomaps that give rise to these columns, and they characterize the entire cerebral cortex. And so we also know that these columns have very specific functions. I've dedicated a lot of my research to studying the function of visual columns in the visual cortex and understanding how they represent different features so that we can decode objects. Another thing we know about columns is that they're connected to other columns very selectively. So here is a picture of an anatomical tracer injection in the central column, and you'll note that each of these are about 200 microns in size, and this tracer labels nearby columns that are of similar functional selectivity. So this picture represents an orientation selective network. While we know this, we do not know what is the organization of the connectivity as one moves from column to column. We don't know if these columnar connections exist at brain-wide scale. Can we map these in vivo, and what is the relationship of these networks to behavior? So to address these questions, I wanted to stimulate a single column and see activation in connected columns, and thereby map the whole network related to that column. And so for this, we investigated different methods using electrical, optogenetic, and infrared neural stimulation. Electrical stimulation is a very traditional method, and it works. However, it's more difficult to keep it confined to a single column due to current spread. Optogenetic stimulation has really revolutionized neuroscience. However, it requires a step of viral transfection, which in the primate brain is not always so reliable. With infrared neural stimulation, one can apply it anywhere in the brain without prior viral preparation. This is a method where a long wavelength light, 1875 nanometers, is pulsed. The pulses are only 0.25 milliseconds wide, and it's pulsed at 200 hertz for half a second. So it occurs like a tiny tickling of the neurons, and the neurons will fire in response to this. And this is shown in the next slide, where you have both excitatory as well as inhibitory responses depending on the parameters of stimulation. We have shown that this stimulation is intensity dependent using optical imaging, where small stimulation from 0.1 joules per centimeter square up to over 1 joule per centimeter square produces an increasing intensity of response. And importantly, you'll see that the light profile here in the lower left corner is very similar to the size of the fiber that we use. So a 200 micron fiber will produce a light distribution 200 or 300 microns in size. So with this method, as well as with the other methods, we have shown that these columns are in very specific networks. So orientation networks and color networks in visual cortex. In somatosensory cortex, we have digit-specific networks. So the next thing we wanted to do was find out whether this could be applied in the MRI to see brain-wide networks, such that stimulation of the yellow column may activate its network of columns, whereas that of the red column may stimulate a different network. So here we use an ultra-high field 7T MRI to achieve the resolution needed to see these very specific units. And I'll make note that it's important to have high spatial resolution for this method. So when we applied this method, we saw that also it produced intensity-dependent bold response. The stimulation increasing from 0.1 joules per centimeter squared to 1 joule per centimeter squared produced increasing bold response, as you can see here on the right. And the bold response is quite reliable. For every single pulse train, we see a nice bold response. So then we applied this to map out different networks in the brain, and today I'll mostly be talking about amygdala. But first, let me just mention our previous paper, which established that this method does work from cortical sites, where we stimulate area 17-18, which has a very well-known anatomy, and its connections are very well studied. And when we obtain the mapping throughout the brain, we see the network that was activated is very similar to the predicted anatomical sites. So this method was shown to be able to map brain connections at mesoscale in vivo. So what about from subcortical sites? Well, for this we chose to study the basal nucleus of the amygdala. The amygdala has many subnuclei. It's an important nucleus for primates because it integrates lots of sensory, cognitive, and limbic and motor information to produce appropriate emotional and social response. And this is important for primates that have complex social behaviors. I'm going to be focusing today on the connections with the insula and with the lateral sulcus. These connections are known anatomically, but it is not known what the organization of connections is within these nuclei. So here I show you a single slice with the fiber optic inserted into the amygdala. You see the signal dropout, the black line from the glass of the fiber optic, and the stimulation site at the fiber tip. And at the right, you see all the slices throughout the brain. And the first thing you'll note that the activations are kind of sparse. So out of 30,000 voxels in the brain, we typically get only 100 voxels activated. So the activations are sparse and highly specific. And I will show you more about that. We then proceeded to decide that we wanted to map out the connectome of the amygdala by penetrating the amygdala at many different sites. And so including the lateral, the basal, the accessory basal, as well as areas CE and ME in the amygdala. And this is a result of one of those experiments in which we stimulated 55 different sites in a single amygdala and obtained a matrix. On the lower row in the matrix, you'll see all the brain sites, brain areas, and on the vertical row are all the sites that were stimulated. So this matrix represents all the connections of one amygdala in the macaque monkey. And so using this matrix, we could compare networks within an individual and also study the topography of connections. And I'm going to show you here an example of topography. This is an example where we penetrated the basal nucleus of the amygdala, shown here with 3 red dots, 3 different sites within the basal nucleus. And we activated the lateral sulcus region of the brain, which includes the insula in red, in green, the somatosensory areas, and in blue, the auditory areas. And you'll see that at the top here, in this top row, there's activations in the insula, as well as in auditory regions, as well as in somatosensory regions. And if you go down to the next stimulation site, a millimeter away, you'll see that the activation has shifted somewhat in this anterior-posterior extent. And you'll see that there's a little more activation in the sensory areas than in the upper row. And as you go down to the next third point, you'll see again the topography has somewhat shifted. While we don't understand at this point the functional meaning of these shifts, that is something to be investigated. Now, we also saw activations in the cingulate from basal nucleus sites. These are 3 examples from 2 monkeys, 3 different hemispheres. And in A, B, and C, you see the stimulation sites, and in the basal nucleus, and in D, E, and F, you see the activation sites in the cingulate gyrus. In particular, in areas 23 and 24, you can see these focal patchy activations going across the cingulate cortex. And you can see this in D, in E, and in F. So 3 different examples. And this correlates well with the motor areas known to be in this motor limbic area, representing hand, face, and body. And so these sites appear to correlate better with the hand and face region, leading us to hypothesize that this basal nucleus site is involved in hand and face motor limbic response. So just to summarize, this one circuit that I've shown you shows that we have hand and face motor circuits involved. We have cognitive circuits in the insular cortex, as well as sensory circuits areas in the auditory and higher order somatosensory areas. So we hypothesize from this that this circuit represents limbic face and hand gesture circuit. And these will provide hypotheses that can be further investigated using other methods, such as electrophysiological and behavioral methods. So just to summarize, INS fMRI is an in vivo method for mapping mesoscale connections, does not require viruses. One can identify the functional node that's being stimulated. It's fast and systematic and can be applied at large scale for connectome studies. We can reveal local and global networks as well. If you have a way to image with sufficient resolution to see laminar definition, you can distinguish feed-forward versus feedback connectivity. And finally, I just want to make one more note, which is the implications of this method for understanding topology in the brain as well as for AI. Previous methods have shown that node-to-node connectivity, functional connectivity exists. Now we have added these arrays of nodes within each of these macroscopic nodes, which changes the mathematics of studying these relationships to array connectivity. And so this is something that I hope the AI people and mathematicians will pay attention to. So finally, I would like to thank my collaborators at Zhejiang University, all the wonderful students and PIs and postdocs who have contributed to this project. My collaborators in the US, and in particular Kathleen Gotthard and Liz Romansky, who worked really hard with us on the amygdala project. And finally, I think my international colleagues who really helped get us started at Zhejiang University. And finally, I thank the funding agencies in China for funding this project. And I thank you all for your attention. Thank you.
Yeah, thank you very much for this great presentation. I also see we already have a question. We will answer it in a second. I just wanted to mention that the presentation will of course be available after the symposiums. There will also be a longer version available, so with longer presentations from the speakers. Just for your information, you can, for example, find it at the Siemens booth. So as I already said, we have a question from the audience, and I will hand that over to Anna directly. Do you think that you can distinguish monosynaptic and desynaptic connections using INS fMRI? And Hiromasa Takemura also wonders if the connection is GABAergic, so suppressive, or do we see negative? So do we see negative fold or not?
Okay, excellent questions. The answer to the first questions, we believe that we can see monosynaptic as well as disynaptic connections. This is based on the evidence that we got, for example, from cat visual cortex, where we got all the predicted monosynaptic connection sites. And in addition, we obtained sites that were not directly connected. For example, sites in the contralateral hemisphere, like contralateral pulvinar, as well as higher order areas like anterior ectoselvian and cingulate visual area. These are not monosynaptically connected to areas 17 and 18. However, they are disynaptically connected. And we could see at the connected site the drop off in the intensity and the correlation with the additional synapse. So it did fall off by an order of roughly half or sometimes fell off more than that at the connected site. And so we can distinguish these. We are trying to do this systematically so that we can distinguish monosynaptic from disynaptic using differences, quantitative differences in amplitude as well as in variability. So I do believe that these can be distinguished. By the third synapse, I believe that the responses are in the noise and we cannot distinguish those. So this method is useful up to a disynaptic extent. And I believe that also with the very low intensity stimulations, one can bias the activations primarily to the monosynaptically connected sites. So that's the way we intend to use the method. Regarding GABAergic response, well, yes, it's difficult for me to answer this directly. One piece of evidence we have is that when we stimulate CE and ME, which is a subnucleus of the amygdala that is known to project to brainstem sites involved, for example, locus coeruleus and Raphe nuclei, that these projections are known to be GABAergic. And so we definitely are able to activate these GABAergic neurons and achieve activation in their target sites. Regarding negative bold, we find that if you stimulate with greater intensity, the response can become negative, and we see this quite consistently, that with increased intensity, not simply with this method, but even with electrical stimulation, that you will get an increased amount of negative bold activations. What this means remains to be established. I've discussed this with other investigators, including Amir Shmoyel, who has focused on negative bold. One possible hypothesis is that in addition to the possibility that you're actually increasing inhibitory responses, another possibility is that there is a temporal delay due to the increased intensity when you're changing the balance between positive and negative aspects of the network. And that is leading to a delay in the bold response, which then will appear as a negative correlation. So those are a couple possibilities about those outcomes. Thank you very much for the questions.
Yeah, thank you very much for answering. And as I don't see a question at the moment, I would suggest to continue. However, if you have any additional questions, then of course post them in the chat and we will address them later on. Okay, thanks Anna for answering these questions.
Thank you.
So let's continue with the next speaker, and the next speaker is Randy Buckner from Harvard University. He unfortunately cannot be with us in person, so but we have also someone from his lab, Ross Mayer is with us, so if you have questions you can also post them in the chat. And now to Randy Buckner. So he will talk about precision estimates of structure and function within the individual. He's with Harvard University. He's the Sosland Family Professor of Psychology and Neuroscience. He's also the director of the Neuroimaging Core at the Center of Brain Science. He's simultaneously affiliated with MGH. He's a neuroscientist in psychiatry and radiology and the director of the Psychiatric Neuroimaging Research Program. His academic career started at Washington University and went all the way to a PhD there. In 1996, he joined Harvard Medical School and MGH for the first time. Then transitioned back to Washington University, and since 2005 he's back at Harvard. He's a fellow of AAAS, the Association for Psychological Science, the Society of Experimental Psychologists, and the American Psychological Association. He holds also different editorial roles, for example, for Cortex, Neuron, and the Journal of Cognitive Neuroscience. He was a treasurer for OHBM in 2001 to 2003, and has also a very large list of peer-reviewed publications, more than 250. And now I'm really glad that he agreed to present in our symposium. Please, Dr. Buckner, we're looking forward to your presentation.
Thank you for having me today. It's a wonderful opportunity to speak at this conference, which I've been attending to since the very beginning. And I want to provide a couple of updates in an area where we've made progress making precision within individual estimates of brain structure, which potentially could allow us to measure change to treatments in individuals over relatively short periods of time. But before I start, I wanted to just step back and actually think about the broad progress of the field. And I'm trying to capture that in just two images here. We've learned a lot about the course of several diseases through the use of biomarkers, including various neuroimaging techniques. And one great success is in the area of Alzheimer's disease, where initiatives, and there are many of them, such as ADNI, or the Alzheimer's Disease Neuroimaging Initiative, have been able to study groups of individuals longitudinally over time with multiple biomarkers and get a good handle of the relative changes in measures as the disease progresses. And that's what's shown over here in an aggregate plot, where we can see that certain measures that some believe reflect the mechanisms of the disease, such as the buildup of amyloid and tau, begin early, well before the clinical manifestations of dementia, which progress late, as shown here in the slide, indicating by the range of normal cognitive function and then progression to MCI or mild cognitive impairment, and then eventually to clinically relevant dementia. In the middle are measures of brain structure, for example, such as the measures of the hippocampus that are intermediate, that begin to change early as neurodegeneration progresses. And in fact, volume of the hippocampus is an excellent marker of neurodegenerative progression in Alzheimer's disease, and related measurements can be made in other forms of dementia. And this is a great success because it tells us as a field what to expect in the progression of a disease, and that can be used to both power clinical trials, but also as endpoints in clinical trials. What I want to talk about today is how we can do better. We know a lot about brain networks in aggregate, shown on the right, and the connectivity of, for example, systems like the medial temporal lobe, the hippocampal memory system, to the rest of the cortex. This is in average groups of subjects. How can we harness the field's understanding and knowledge about the progression of disease to make better biomarkers, better measurements, more precise measurements, to ultimately fuel the next generation of translational efforts? And so thinking about this, I want to focus today's talk on one broad question. What if we could quantify changes in brain anatomy so precisely that a treatment effect could be detected within a single person? Now, this would open up a number of possibilities. In general, I think we'd have more precision in any studies we're doing in groups if we were able to harness that level of individual precision. But we might also be able to imagine tackling the earliest stages of clinical translational development. For example, in early phase 1 clinical trials or safety trials, where relatively small numbers of individuals are receiving treatments, and be able to detect their effects on neurodegeneration in the early phase of development. And it's interesting and timely here in the United States, there's provisional approval for one of the first disease-modifying therapies from Biogen. But one of the challenges in that development is that while that therapy is known to affect amyloid deposition, to affect correlates of Alzheimer's disease, there really isn't yet robust evidence that it's changing the course of neurodegeneration, or that it's having large efficacy effects. What if we were able to make measurements early in the development of drugs in small numbers of individuals to see if there was efficacy, if the trajectory in neurodegeneration was changing? So turning to Alzheimer's disease, we know a lot about the disease. We know that brain atrophy, in general, at the end stages, this tremendous atrophy is shown here on the left in a cartoon example. But if you use MRI to go in and see that atrophy, you can see here to your naked eye the changes over a little bit less than a decade from an image that Cliff Jack at the Mayo Clinic was able to provide. You can actually see to your own eyes, without any quantitation, the shrinking of the hippocampal formation as the disease progresses. And so we have clear targets for measurements, and we need to make them more precisely. In the evolution of measurements in disease, it has progressed from manual measurements, as shown to the left, where individuals were hand drawing cortical or subcortical regions such as the hippocampus, to progressively more automated variants. And this was a big breakthrough, such as shown here, an automated segmentation of the hippocampus in the tool FreeSurfer that many in our community have used. Now that we can make automated measurements, it allows us to scale up and actually see the effects of improvements in acquisition parameters very directly. And automation leads to our ability to scale things up. For example, in the UK Biobank, here is a plot made by Jingnan Du in over 30,000 individuals over the age span that shows you the distributions, the volume of the hippocampus. I just want you to focus on the lines in between that are separated for men and women. And what you can see is that you can see two things. First, that men have larger hippocampi than women. There's a general head size difference between men and women, but also in all of us, there's a decline in hippocampal volume with age. In Alzheimer's disease, that decline accelerates markedly. So how can we make such measurements, not in 30,000 individuals, but in small numbers, such as can we estimate volume change in an N01 with extreme precision? Now, there are a couple strategies to do this. The first is to push the limits of resolution, to get more advanced acquisitions and different kind of acquisitions that could stably estimate potentially subfields or small changes in volume, as shown here in one demonstration from Susan Buckheimer and colleagues using a turbo spin echo sequence, where the Human Connectome project acquired hippocampal sections with 400 micron resolution. You can see here the exquisite resolution that one can achieve. There's a challenge to increasing resolution in longitudinal studies because the exact position in the error of the measurement between the times and head positions will be the error term barrier that one needs to overcome. There's another strategy that one can use to increase precision that I want to talk about for the remainder of the time because it's less appreciated, which is rather than getting better images at higher resolution with longer acquisitions, to actually go in the opposite direction, to acquire images faster and acquire more of them, to get more precision by averaging over multiple images and to do so rapidly at low burden. And so in that arena, we need to push the limits of speed. And so here is an acquisition circa 2008, a typical T1 structural acquisition where you're seeing here segmented. This one takes about 6 minutes. And here is an acquisition in 2017 accomplished with a wave Kaipi strategy where you can accelerate and get a robust image in 1 minute. Now, there's a couple of things you'll notice about this. To first view, the image isn't quite as beautiful. The contrast, the signal to noise isn't quite what we got in the typical acquisitions. But when you push these acquisitions through standard automated measurements, such as in FreeSurfer, you can see that the cortical surface can be reconstructed from these faster images in nearly the same way. Now, another approach that we've been collaborating on with Siemens on is to use compressed sensing techniques, which effectively, rather than using standard accelerations, use radial coverage and incoherent undersampling to sparsely sample the image and then use machine learning techniques to reconstruct the image back. These have been remarkably successful as well. So shown here is a conventional T1 MP RAGE image on the left. And here on the right is now a compressed sensing variant with its segmentation taken a little over a minute. And as you can see, you get nearly the same results. And I want to quantify that for you rather than just say that qualitatively. Here is a plot of hippocampal volume in 16 individuals, where within the individual, we required a standard 6-minute MP RAGE. And we also acquired a fast compressed sensing variant, such as the one you see to the right. And you can see that there's near perfect correlation between the two. That is, if an individual is measured to have a larger hippocampus with the standard acquisition, they also had a larger hippocampus with a fast acquisition. Almost all of the variance, the size variability between individuals was retained, even though the acquisition here took 1/6 the amount of time. But there was a subtle catch that we noticed, and this was noted by Lindsay Hanford, she used a very interesting procedure to actually plot, so that to see this, which I'm showing you here on the left, what you're seeing is actually 6 independent acquisitions and the cortical surface computed from the 6 acquisitions on top of each other. If you look in parietal cortex where the red arrow is pointing, you can't even tell that there are 6 different lines because they give the estimates of the cortical outer boundary and the inner boundary are so similar that the line falls on top of one another. But if you go to another part here in the temporal lobe, near regions of traditional susceptibility artifact, something that we've known about for a long time for our bold acquisitions, you can see that there's a systematic bias in all of the compressed sensing variants. So we dug into that with the help of Siemens a little bit more. And what we found is, and it was unexpected, that a change in the acquisition between the standard sequential acquisition and in the incoherent undersampling, the compressed sensing versions, led to an artifact. And Tom Hilbert at Siemens played around with this and suggested that if we switch the acquisition, which in practice meant from going from a sagittal, or traditional sagittal acquisitions, to a coronal acquisition, we might be able to overcome even this bias. And when we tested that, as shown below, that in fact was able to recover even these difficult to image areas. And so on the left here, we're showing the traditional acquisition estimate and the artifact that is present when you acquire sagittally, and all the way to the right, you can see that that artifact is overcome by a simple switch. And this now even takes this robust method and even is able to recover some of the troubling areas with a fast acquisition. So to show you the potential of these methods, I want to turn back to now what we need to measure in Alzheimer's disease. As we started off with, we know that there's atrophy in the Alzheimer's brain broadly, but in the hippocampus specifically. Now I'm showing you to the right what you can achieve if you exploit this, push the limits of speed, of fast scanning. So here is a plot of the measurement error when you use automated procedures to reconstruct the volume of the hippocampus. And rather than acquiring one scan as traditional, you require many fast scans. And what's being plotted across the number of scans is the absolute error in measurement. And as you can see, in a typical individual, there's 6 individuals plotted here, lines for 6 individuals. You might, in a traditional measurement, get errors of 1, 3, or 4%. As you repeat the measurement and pool the average, the test-retest measurement error can drop to fractions of a percentage, in some individuals as small as 0.3%. This places us in an arena where we may be able to now get precision measurements, if not in one, in small numbers of individuals. One thing that has struck me about our translational trajectory is that there are many places, including in the recent trials that look like they may be successful to some degree, is that some individuals are benefiting from these therapies and others not. Here we may have a way to make precision measurements to even explore that arena, because we can have measurements with known properties in individuals to even be able to pull out the subsets of individuals who are benefiting from therapies, even if there are many others who are not. So looking into the proverbial crystal ball into the future, I imagine a world where we're able to get better measurements in individuals, and that will further generally the translational objectives, but even more broadly, that going fast in acquisitions might become the norm. And just to throw out sort of an imaginary target, I wonder what we can do if we push along this arena and start to target something like a 5-minute neuro battery. The entirety of the battery from start to finish. That is now imaginable when you can see things like a T1 structural image taken here with a compressed sensing variant that's highly accelerated in 43 seconds. What can we do if we push the limits of this in multiple acquisitions? Can we get to something like a 5-minute neuro battery? And so with that, I'd like to end and thank my many collaborators. In particular, I want to call out Lindsay Hanford, Ross Mayer, and Jared Nielsen, who have spearheaded this work on our end, but also our collaborators, Siemens, Tom Hilbert, and Tobias Kober, who have been instrumental in helping us to push forward with these compressed sensing variants. Thank you for your time.
Yeah, thank you, Dr. Buckner, for that great presentation. And we have Ross Mayer from Randy Buckner's lab with us. So in case you have any questions, just let us know. As a starter, I have for sure a question. So, I mean, it is very fascinating to think about the possibility to really lower the error bar to a level that you can see these changes in an individual. Now, we heard this is not yet feasible. And the question would be to Ross, from your perspective, what do you think? What is still missing? Do we need, I don't know, a fast scan with higher resolution now, or what is still missing in order to, get down to these measurements in an individual?
So the approach we were taking here was, first of all, to not to average the images together, but to treat each very rapid image separately through FreeSurfer, and then work on averaging of the resulting morphometric numbers that came out, the cortical thickness for each region and volumes of structures and so on. So in terms of going forward from here, certainly being able to apply these methods, we're still working out some kinks that we're seeing with the MP range, as Randy explained, but being able to apply these methods to all the regular clinical structural modalities, T2 weighted, flare scans and so on, so that we can accelerate anything, any type of scan. But also then, I don't know if we're now going to push the limits of the analysis of these images and the detail that you can extract using FreeSurfer or some other automated tool, possibly something that involves more of a machine learning approach, which can improve on the certainty of those things as well.
Okay, thank you. And I see while we were talking, there came a question, and it goes into the topic of the averaging. So when you average structural data from the same individual many times, what is the recommended procedure for averaging? So do you simply average all images, or do you perform some kind of registration, assuming that motion occurred between the scans?
So yeah, just to reiterate that point, we're not averaging the images together. We consider each one separately. The plots that Randy showed, where the different cortical tracings from each different scan are layered over the top of one another. That particular example, amazingly enough, came without having to register the images together. That subject was so incredibly still in the magnet. And the tracings just came out that way. So that is kind of an unusual example. We did, in the general analysis routine, we did start by registering all of the images together, just to put them in a common space. So sort of taking one part of the FreeSurfer longitudinal process, but not using the rest of it. Lindsay, who's my colleague who's done most of the analysis on this, has a detailed poster that's being presented tomorrow, 12 p.m. US Eastern Standard Time, number 2722, where she can talk in more detail about this. I think now she's adopted the actual FreeSurfer longitudinal stream to process all of the images separately. So in that case, yes, they are all co-registered before using the FreeSurfer processing tool.
Okay, thank you very much for that detailed answer. I have maybe one final question. I mean, we heard about different acceleration techniques. So compressed sensing was mentioned, and also wave Kaipi. Do you see? Do you think in terms of speed, this is now fast enough, and we can now more look into the resolution piece, or would you still think faster is still better? And for example, using new techniques like, I mean, the deep learning is all over the place, and maybe that brings us into close to that goal.
So from the MR images themselves that we've acquired, going to a compressed sensing factor of about 10 really does start to lead to noticeable degradation in image quality. 8 is certainly lower than you would expect to see, but it still gives you a very high quality scan that's radiologists can read, I think, without any complaints. But at a factor of 10, it's really a problem. Although as the cortical surface tracings show, free surfer can even handle that. I wouldn't go out on a limb and say it would handle every single times 10 accelerated scan we give it, but at least for these examples, it was able to. So I think that the tools have a lot more capability in them than some people have given them credit for. But I just lost my train of thought. Sorry, can you repeat the second half of the question?
Yeah, no. I was just thinking about if there's enough. So if it's already fast enough, if we should look more into the resolution piece now, or if the paradigm with the faster the better is still valid.
Yeah. So from that point I was making, we may have reached the point, at least with current techniques, where fast enough is enough. So yes, there's the chance then to look into the resolution question. And again, I think the quality of the tools, software and image processing tools that we're working with, if they could potentially, handle an image that's 20 times undersampled, it may give us something on the computer screen of the scanner console that we can barely recognize as a T1 weighted image. But if the image processing tools with some machine learning capability in the background know what it's looking for, then maybe that would be usable enough, and then we could whip through every one of these scans in 30 seconds or something. But I think that's still some way off at this point.
Yeah. All right. Thank you for your answer again. And I see we're on top of the hour, and that basically also concludes that symposium. So I want to thank again the speakers, and also Anna and Ross for being available in this symposium here to answer your questions. Thank you to the audience for attending. Thank you for also for your questions. And if you have further questions, please reach out to us at the Siemens booth. We are happy to forward any questions. And yeah, I would say thank you very much again, and enjoy OHBM 2021. And hopefully we can see each other next year in a live congress again in Glasgow. So thanks again, and have a good rest of your day. Bye-bye.
Thank you.
Thank you, Andrew.